Welcome, buenos dias. We are very excited to hear this interesting talk, Unveiling the Immune Dance, Macrophages' Role in Type 1 Diabetes, Immunotherapy, coming to us from Spain, in just outside of Barcelona at the IGTP. We have Dr. David Perna-Barrull, and he's a doctor. He has a doctorate in advanced immunology with a special award from the Autonomous University of Barcelona, UAB. A master in advanced immunology and a degree in biotechnology from the UAB. He's a postdoctoral researcher since 2021 in the Immunology of Diabetes unit, led by Dr. Marta Vives P. at the German IGTP in Barcelona. Since 2016, he's participated as a researcher in projects funded by national and international agencies. He also has had a time researching at the Department of Immunology at the University of Hamburg, Germany. His lines of research focus on the study of the influence of prenatal environment factors such as drugs on the development of type 1 diabetes, defining new biomarkers for the determination of the partial remission phase in pediatric subjects with type 1 diabetes. And recently, he is highly interested and focused on the role of macrophages in new immunotherapies targeted against type 1 diabetes. All of these projects resulted in 17 original articles, and he's a member of the research group of the government of Catalonia, Advanced Immunotherapies for Autoimmunity, and the member of the working group Basic Research in Diabetes from Sociedad Española de Diabetes. Welcome, David, and thanks so much for joining us.
Okay, thank you for your kind introduction, Monica. I'm delighted to be here to be able to present our work in this forum. Today I will show you one of our last works, and that's that talk about macrophages and the role in type 1 diabetes and immunotherapies. First of all, I would like to present our group. We are the Immunology of Diabetes unit. We work in the German Trias Pujol Research Institute in Badalona, in Spain. We are a group of scientists and medical doctors that we work together. In the center, we have Ivan Garcia Loza, is the master student that together with me, we performed most of the experiments for this work. Okay, as I say, we work here in the German Pujol Research Institute. We are near the hospital with the same name in the Canruti campus. This is the mountain of Badalona. And to put you on place, we are quite near Barcelona. And as you can see in this image, we have a really nice view of the Barcelona from our workplace. Okay, the main research lines of our group are, as Monica said, the pathogenic mechanism of autoimmunity in type 1 diabetes, the pediatric type 1 diabetes tolerance, spontaneous remission, and biomarker. I think this line is super interesting because we are looking for these biomarkers that can determine this spontaneous partial remission, that it's a good moment to perform immunotherapies or to well, it's quite interesting for these pediatric patients. And our third line is the immunotherapies for the prevention and treatment of autoimmune diseases. The article I'm presenting today, it's inside this third line. Okay, this is the article we published last May in the Journal of Autoimmunity. The article is called Targeting Macrophages with Phosphatidylserine-Rich Liposomes as a Potential Antigen-Specific Immunotherapy for Type 1 Diabetes. We'll start with a brief introduction. Then I will show you some of the results we obtained, and I hope we have a fruitful discussion after my talk. Okay, as you may know, type 1 diabetes is an autoimmune disease caused by the destruction of pancreatic beta cells by T-CD8 lymphocytes. This disease has a big problem for the study, is this long asymptomatic period. Well, this period can be divided into stages. In the stage 1, we have this break of the tolerance and the reduction of the beta cell mass. In the stage 2, patients suffer from dysglycemia, but they don't need this exogenous insulin. It's when the beta cell mass is sufficiently reduced that the patient has the symptoms of type 1 diabetes and the hyperglycemia that we start the insulin treatment with the diagnosis of the disease. Most of the patients, especially on the pediatric population, has this partial remission phase where the glycemic control improves a lot. In the long-standing disease, we think that there are some wave of partial remission, but we don't know how to detect. And for this, the importance to study the biomarkers of this partial remission to try to detect this wave and improve the therapies that can provide to the patients. Okay, we are fighting against a disease that has neither a cure nor prevention. Okay, but how this disease start? Well, we have a triggering injury that will cause the apoptosis of some beta cells. In people without the disease, these apoptotic beta cells will be efferocyted by macrophages and dendritic cells. This gives them a tolerant signaling against the pancreatic beta cells, maintaining a healthy isolate. However, people that will develop the disease have some problems in this pathway, and the apoptotic beta cells will become necrotic. When a necrotic cell is captured or phagocyted by dendritic cells or macrophages, they start an inflammation process releasing the pro-inflammatory cytokines. And later on, the same cells will do an antigen presentation to T lymphocytes, and this will mark the starting of the autoimmune attack against pancreatic beta cells. Okay, if we want to cure this disease, we aim to promote this path of tolerance. At the same time, we have to avoid this necrosis path. In this sense, our group designed a particle that are quite similar or that trying to mimic these apoptotic beta cells. This particle has on the membrane phosphatidylserine that gives them the ADME signal or to the phagocytosis, and in the inside we put antigens for the autoimmune disease we are working on. In this case, we can put insulin antigens to work with type 1 diabetes. What is happening with this particle is that we are mimicking the efferocytosis. Then dendritic cells eat the liposomes, and these liposomes give the dendritic cells a tolerogenic signaling. This will stop the autoreactive CD8 T cells and increase or expand the T regulatory cells. This idea now is licensed by a health therapeutic that is using these liposomes as a platform, not only for type 1 diabetes, but to reestablish the tolerance in different autoimmune diseases. And here you can see the pipeline of the health therapeutic company. Okay, we have been working with this liposome for quite some time now. We started, one of the first articles we published was in 2015, and we checked that these liposomes can arrest the autoimmunity in type 1 diabetes in the animal model. And this was a good starting point because we know that we can, we have this stop of the autoimmunity, and we want to check what happens with human dendritic cells. And after giving the liposomes to human dendritic cells, we observe that they promote tolerogenic features on these cells, and they have this similar mechanism of action that what we found in the mice, and we find similar results when we use pediatric patient cells, and we has this the same tolerogenic signaling. And that's good because we have a therapy that can be used in both population, in pediatric and adults, and that's quite interesting. Okay, but we have studied a lot about dendritic cells, but what about macrophages? Because at least us, we tend to forget them, but they are essential phagocytes in the maintenance of tolerance to self. Then the aim of the study, we think that's the moment to deepen into the role of macrophages in the mechanism of action of PS liposomes for type 1 diabetes and study potential synergies of PS liposome therapies with other drugs. Okay, what we know about macrophages? Well, there are an innate immune system cell. There's tissue resident cells normally, and they are known because they are quite the good phagocytes and as a scavenger cell that eat everything that reach the well. Um, these macrophages are important for the inflammation, but not only for them. For that, if they are important for the resolution of inflammation. They are typically classified as M1 or classically activated or inflammatory macrophages and M2 or alternative activated macrophages. However, when we talk about macrophages, I think it's better to think them as an spectrum. Their phenotype should be it's more like an spectrum because they have a high plasticity. Then the same macrophage can change from one side to another side, or we can find some phenotypes in the middle between a classically activated macrophage and a regulatory macrophage. Wherever we know that there are some cytokines that can polarize the macrophages, for example, LPS and interferon gamma will generate a classically activated macrophage. Okay. In type 1 diabetes, they are quite important because they are one of the earliest cells to infiltrate into the islets. They promote the lymphocyte recruitment and secrete high amounts of IL-1 beta and TNF alpha. And some people have reported that subjects or patients with type 1 diabetes has defects in the differentiation to M2 phenotype. Then, if this macrophage can do the switch from M1 to M2, we have a continuous inflammation that's very dangerous for the pancreatic beta cells. However, we can eliminate these macrophages from the body because they are quite important or involved in the pancreas and beta cell regeneration. Then, if we can um, modify them or help them to change again to M2, it could be very interesting for a future cure on type 1 diabetes. Okay, as I said, now we show you some of the results. First of all, we wanted to check that if our therapy, our liposomes are captured or phagocyted by macrophages. And for this, we use an animal model. In this case, we use the non-obese diabetic mouse, and we administer with fluorescence liposomes. After 15 minutes or 6 hours, we take the spleen and the liver and obtain the immune system cells from these two organs. And we analyze them using flow cytometry to see which cells has more phagocyted the liposome. We observed that some cells in the spleen and in the liver has the ability to phagocyte and capture our liposomes. And when we look which cells are the best phagocytes, we observe that in the spleen, macrophages has the most percentage of phagocytosis, reaching almost 30-40% of the macrophages has engulfed some liposomes particles. But in the liver, we have similar results. The macrophages were the cells that has more phagocytosis, and it's almost 100% of the macrophages uh, have captured some liposomes. Then we now have the evidence that when we are doing our therapy or when we are injecting our therapy, macrophages are quite important because they are phagocyting a lot of our particles. Then will be a good idea to look what our liposomes are inducing on macrophages and if there is some change on them. And in this case, we go to the human approach and we use an in vitro experiments from monocyte-derived macrophages, and to analyze the effects of liposomes, we check the capacity of macrophages to phagocyte these liposomes and the changes in the phenotype, in the cytokine secretion, and how it changes the ability to stimulate T lymphocytes. Okay, first of all, to generate the macrophages, we take blood from subjects with type 1 diabetes or controls. We isolate the PBMCs using a FICOL, and then we obtain the monocytes using a CD14 isolation with a magnet. These monocytes were cultured for 6 days with MCSF, and at day 6, we use a polarization stimuli to obtain these 4 different subsets of macrophages. That's the subset we will work in all the paper. Okay. Um, well, this is the clinical data from the participants in the study. We didn't observe any significant difference between control subjects and type 1 diabetes subjects, and the subject with type 1 diabetes has a normal glycemic control and no secondary complications. Okay, first of all was to check if these macrophages internalize our particle or liposome and it's not stuck on the membrane. And to do that, we use the confocal microscopy. And if we take a closer look in these M1 macrophages, we can clearly see in blue the nuclei, in red the actin that marks the membrane or the cytoplasm, and in green our liposomes. And we can see here in the center of the cell that we have some liposomes inside, and on the membrane we can observe that there are some liposomes that has been just recently engulfed, and they are trying to move again to internalize these liposomes. Okay, we know that they're internalized, but we don't know which of these four subtypes of macrophages are the one that has more affinity for our liposomes. And we do a time course phagocytosis assay with we analyze using flow cytometry. What we obtained is that the M1 macrophages is the most phagocytic subset, followed by M2C, and then followed by M0 and M2A. The interesting thing of this graph is when we compare the capture capacity of control patients, control subjects, and type 1 diabetes subjects, we didn't find any difference. And that's really good because we know that there's anything, well, our therapy could be used with the patients without any problem. Okay, now that we know that the macrophages can internalize and there are quite good uh, well, they can capture easily these liposomes. We want to check if our liposomes can affect the viability of our macrophages. To do that, we use an IncuSite, that's an incubator with a fluorescence camera, and we put together the macrophages with the liposomes and with propidium iodide to check for the mortality. As you can see in the red line, we have the positive control that we put sodium aside on the culture, and we can observe this steady increase on the propidium iodide intensity. However, we didn't see this increase when we put our macrophages with two different concentrations of liposomes or without treatment. Then we can conclude that we have low mortality in the first 72 hours when adding liposomes to the different macrophage subtypes. Then that these liposomes are well tolerated by our macrophages. Okay, now the interesting part, that's what's happening to the phenotype of these macrophages when we are using liposomes. We culture again these macrophages, we obtain these four different subsets, and we put together with liposomes for 48 hours. To analyze the phenotype, we use this full spectrum cytometry because macrophages has a high autofluorescence, and it was quite easy to take out of the analysis the autofluorescence using this type of cytometry more than if you use a conventional cytometry. Okay. First, we check if we have some critical difference between control subjects and subjects with type 1 diabetes. As you can see in the heat map, the profiles are quite similar between the two groups. And however, we find some alterations when we compare between them. For example, in the M1, we find some differences, some alterations in CD80, CD86, and HLDR, especially in these four patients that we want to study a little bit more about them, but we have to do it in a future. In the M2A, we have a difference, alterations in CD86, HLDR, and CD200 receptor. And in the M2C, we have alterations in CD80 and CD163 when we compare the two groups. But okay, it's quite interesting, but that's not the we are doing this study. We want to compare what happened after liposome treatment. And here we have the data. We have quite a lot of data, but I will try to explain the best I can. First, on the left side, we have the control subjects. In the right side, we have the subjects with type 1 diabetes. And when we compare the different subsets, in the M0 subset, we found an increase of CD86, CD163, and CD36 in subjects with type 1 diabetes, and a decrease of PDL1, PDL2, and CD206 in the same subjects when we treat these macrophages with our liposomes. In the M1 subset, we didn't find any significant change due to the liposome capture. In the M2A, we find an increase in HLA-DR in the control subjects and a decrease in the CD200 receptor in the subjects with type 1 diabetes. And in the M2C, we find an increase of CD86, HLA-DR, CD163, and CD36, and a decrease of CD206, MERTK, and CD14. Okay, there was we find some quite we find some alterations. The most interesting parts, the M0 and the M2C, that we observe that our liposomes can produce some polarization of these macrophages. Then I think they are doing an interesting effect, but we will see what happened with the cytokine release. In this case, if you remember, the M1, we didn't find any difference in the phenotype after the liposome capture, but we have found a reduction of inflammatory cytokines after the treatment with liposomes. And we find a reduction of IL-6, IL-12, IL-23, and IL-1 beta, but only in the control subjects. And we find too a reduction of gas-6, and that's quite interesting because this gas-6 is a molecule, a soluble molecule that binds phosphatidylserine and helps the efferocytosis of these particles by other cells. And if we see this reduction after the treatment with liposomes, I think that we can think that the liposome is binding to gas-6, and this helps the liposome to be internalized. Moreover, when we compare between control subjects and subjects with type 1 diabetes, we find some difference in some of these cytokines. One is a reduction on gas-6 expression, a tend to reduce IDO, and a tendency to reduce the TGF-beta when we compare these two groups. When we look at the cytokine secretion of M2C, we didn't find any difference, and we find similar results in the M0. There's no difference in the cytokine secretion after liposome treatment. It's true that some of the cytokines we check here are much more secreted in M1, and I think that could be one of the reasons that we didn't find this difference in after liposome treatment. But on the other side, it's good that we don't see an increase of these pro-inflammatory cytokines, and that's a good result for us too. Finally, we want to check the functionality of these macrophages. For this, we do a proliferation assay. We pulse the macrophages with insulin peptides, and we put them together with T cells from patients with type 1 diabetes to see if macrophages can induce an antigen specific proliferation of T lymphocytes. And first of all, what we observe is only M1 macrophages can do this can induce the proliferation of antigen specific CD4 and CD8 lymphocyte. And the interesting part is when we treat these macrophages with the liposomes, we see a clear tendency to reduce the proliferation of both populations. And that's quite good, and it confirms that liposomes are giving our macrophages tolerogenic signaling, and they are stopping the autoimmune attack. Okay, Then the take-home message is that macrophages are key capturers of tolerogenic nanoparticles, even in subjects with type 1 diabetes, that PS liposomes are efficiently phagocyted and well tolerated by macrophages, then that our thinking supports the benefit of PS liposomes for inducing immune tolerance in type 1 diabetes. Finally, I would like to show the graphical abstract because it's a um, good view of the results on the paper, and we are quite happy with how this image was made. And sorry. Finally, I want to acknowledge all the people that work in this project, and to all the people that have given us some blood for the experiment. That sometimes this is the most difficult part to obtain the samples to do our experiments. And thanks for your attention. And if you have any question, I will be glad to ask uh, wherever I can.
Thank you, Dave. This was really an interesting talk. We do have some questions. The first one's from Katie Gerder from Boston. Do the differences in the M2 phenotype in the macrophage correlate to disease duration? And does a decrease in cytokine levels have any impact on their HbA1c? You can
Okay.
see it in the chat if you want to read it out yourself.
Yeah, if you can repeat, let
Sure.
me.
The first question is, do the differences in the M2 phenotype in the macrophage correlate to disease duration? Oops, I'm not sure what's happening.
Uh, sorry, I think I closed the share. I'm sorry. No, it doesn't correlate with.
Maybe go back to your screen.
Yeah.
There you go. That's better.
Well, about the difference in the M2 phenotype, no, it doesn't correlate with the disease duration, because we have from people that has like 2, 3, 4 years of duration of the disease until 16, 20 or more years, and when we analyzed, we didn't find any correlation with the disease duration.
Okay, great. And how about this? Does a decrease in cytokine levels have any impact on their HbA1c?
Mm, was thinking, I think it's interesting. I think we analyze not directly the HbA1c. I think there are some cytokines that correlate with their levels, but not all of them.
Okay. I wanted,
Sorry,
that's all right. Don't worry. I know you're doing
it's
great.
quite a
Just with the
specific question.
language. Yeah, it's fine. I had a question with one of the earlier slides. You showed like a sort of a marked increase in the liver uptake of the liposomes, correct?
Mhm.
And could that be attributed to the presence of the Kupffer cells in the liver, or could you say that's an off-target effect, or is it, just it doesn't matter?
No, I think that's um, principally due to the Kupffer cells, because
Mhm,
they are, well, um, the liver is quite important for tolerance,
mhm,
and we are quite happy that we have this high phagocytosis on the liver, on the Kupffer cells, because as I said, that they are important for the tolerance. Then yeah, I think the Kupffer cells are really important in that we have this well, they're capturing the liposomes. The problem is a little bit more difficult to study in human, the Kupffer cells, than in the mice. And for this, we try to do this in vitro approach using monocyte-derived macrophages. We know that they are not exactly like Kupffer cells, but to extrapolate some of the results could be interesting. But yeah, I think Kupffer cells are the ones that are having this high phagocytosis of the liposomes. That's one of the results that we were looking for when we do the experiment in the mice.
Great. So you're happy with that outcome? Good. Okay, and then I just wanted to ask, open it up to
Um,
one or two more questions. There's a bunch of people in the audience that probably have some good questions. Anyone at all?
yeah.
Um,
Okay.
okay, last call for questions.
Hey,
You lied.
Monica, there are a few
Okay.
questions in the chat.
Oh, yes,
Yeah.
Oh, here are a couple that were above. Sorry about that. Here's a couple questions. Why are liposomes, this is Kevin Bodie, why are liposomes treated macrophages reducing CD4 T cell proliferation? What's the mechanism here? Have you used autoantigen containing liposomes?
Yeah. Sorry, I should have explained that better. Yeah, we use in that last experiment when we pulse the macrophages with the insulin. Then when we put the liposomes, we use liposomes with the insulin antigen inside the liposome. Then uh, I think we are generating more regulatory macrophages, or there are less. And producing less cytokines, and they have more difficulties to stimulate the CD4 lymphocytes. But yeah, we use an antigen and insulin inside. We put insulin, the antigen of insulin, inside the liposome to do that experiment.
Okay. And then this is another question from Kiati. Have you planned to study the effects of these liposomes in autoantibody positive individuals?
Well, when we use patients' samples, they are always autoantibody positive.
Hmm.
It's one of our checkpoints before using a patient. That we know that they are, most of them, or not all of them, are autoantibody positive.
Okay. And then is there any differentiation between those who have one or two autoantibodies, in sort of their progression to type 1 diabetes? Are you seeing any differences in effects?
No. We didn't find any difference. I think wherever you are clinically diagnosed with the disease, the number of autoantibodies, it doesn't matter. I think
Okay.
this in the pediatric population could have some effect, but when we study in the adult, we didn't see any difference because they have one or two or three autoantibodies positive.
Got it.
And you know that normally the autoantibodies with the time in some patients disappear or change from EA2 to GAT to insulin. That's, I think that we didn't see an effect due to that.
Mhm. Great. Well, that's good news. Well, I think we're sort of at the end of our time here. Thanks for this great presentation. Thanks for all the questions. And I hope you have a great rest of the day, and we'll look forward to seeing what comes next from the laboratory.
Okay, thank you for the opportunity to be here, and I hope we can present more things in the future.
You got some clapping in the audience, especially from Bruce for Cherry. So that's fantastic. Have a great rest of your day, David. Thanks for talking. Bye-bye.
Thanks. Bye.